Okay, hi friends. Today I want to talk about just an overview of the endocannabinoid system, reviewing some topics that can be incredibly confusing when talking about the ECS, and just reviewing the different parts of it that interplay to cause the physiological effects that we feel when we ingest cannabis of some sort. So the first thing that I would like to talk about is the concept of ligands. So this is often kind of a very vague concept when we're talking about ligands because they could be anything from light to temperature, but in most cases, in this course, we're going to be talking about small molecules. So when we think of ligands and we think of our receptors, I'm going to draw a transmembrane receptor here. So that means it is spanning through the membranes. And we'll go more into the structure in a minute of transmembrane receptors. So this is our lipid bilayer, that's our receptor. When we think of ligands, I think a synonymous term here is key. And what exactly does that mean? It means we're referring to the reference of the lock and key for activating receptors. So in this case, your ligand, say it's a small molecule, is going to be your key. So say it's CBD is your key, then your lock would be your CB2 receptor. To activate this receptor, we would need a specific key to go into a specific spot in that receptor and cause some sort of downstream effect. We'll often see ligands kind of referred to as these shapes that fit perfectly into a specific receptor. But just keep in mind when we draw something like this, we're actually referring to something like this. That's going to perfectly interact with this receptor. Maybe this hydroxyl group will hit a specific part in that receptor, and that's part of the key mechanism to cause those downstream effects. So some ligand examples when we're talking about cannabis. Would be anandamide, 2-AG, THC, and CBD. So these you could kind of class into two separate categories. The first would be your endogenous. Endogenous ligands, meaning endo, they're coming from inside your body. Your body is actually making these molecules from other things that are in your body. And then you could call these your exogenous ligands, exo meaning from outside of the body, or just your phytocannabinoids and your endocannabinoids. So phyto meaning coming from a plant, endo meaning coming from inside of your body. So again, it's called your endocannabinoid system because it's inside your body and you are making endogenous compounds to activate your endogenous receptors. So just a kind of fun fact of how these receive their name. So many times we will traditionally use a plant, say it's cannabis or opium. So we used cannabis, we found that it caused some sort of biological response. Then scientists said, I want to figure out why that response happened. So then they discover the receptors in our body. So the cannabinoid 1 and the cannabinoid 2 receptor. And then we discovered that there's actually molecules in our body that are meant to hit that receptor. So those are your endocannabinoids. But the discovery was possible because of cannabis. So the whole system is named after cannabis, even though that system is still existing and operating without cannabis present. A similar story is for opium poppy. So we have used opium as humans for thousands of years. We didn't know that our body produced small little peptide molecules that were able to activate those same receptors. So then when we discovered that system, we called these molecules the endorphins or the endogenous morphines. So that's just a fun fact. But what's really important here is that when we say endo, we're talking about in our body. And when we say phyto, we're typically meaning plants, but it's really any photosynthetic organism. All right, so the next topic in the endocannabinoid system I'd like to talk about is receptors. So ligands activate receptors. So again, our small molecule is going to bind to some special part in our body where it's going to cause a downstream effect. We have so many different kinds of receptors in our body. Some of them are activated by light, some are activated by temperature or heat, some are activated by pressure, and then some by chemicals. So when we're talking about the endocannabinoid system, we're talking about one specific class of receptors, mostly. And these are GPCRs. And that's G protein coupled receptors. So GPCRs are unique to eukaryotic organisms. So something like a bacteria would not contain GPCRs. Humans have over 1,000 GPCRs and they have different roles throughout our body. Another name for GPCRs is 7 transmembrane receptors or 7 transmembrane helices. So the receptor, again, this is our lipid bilayer that makes up our membranes in our body. And then if you remember from biochemistry or just Google that different protein structures will change. So you have your primary, secondary, tertiary, and quaternary. So the structure of an alpha helices or an alpha helix is kind of looks like DNA, but it's swirled like this. So a 7 transmembrane receptor has 7 alpha helices that are going to go through the membrane. So again, these are transmembrane receptors. They're going through the membrane. 1, 2, 3, 4, 5, 6, 7. So what you'll notice in this diagram, which is very important to the function of GPCRs, is that you have one part that's on the outside of the cell, and then the other part is in the inside of the cell. So this is how these receptors are able to bind to specific ligands. And then on the outside of the cell, the ligand binds on the outside of the cell and then causes a change which will facilitate some change on the inside of the cell. So these are really important messenger molecules. So this will be the only time I'm drawing in these alpha helices, but you'll also see these simply drawn like this. Dr. Dankos has showed a few examples where the GPCR looks like this. So let's delve into the biochemistry a little bit more and why these receptors are so important. So just to reiterate, our CB1 and CB2 receptors are GPCRs. They're in this class of receptors called G protein coupled receptors. Okay, So again, the name, G protein, coupled receptor. We know that this is the receptor. So this is the G protein. It has 3 different subunits of this protein, and it is also bound to a molecule called GDP. So when it is bound to GDP, it is not active. So you can see that this has kind of a V shape for the receptor part here, and the ligands present are circles. So this is not being activated. The subunit is bound to GDP. It is not activating any sort of response in the body. It's simply just existing on the cell surface. So what happens when something in our body changes? So let's say, for instance, you smoke marijuana or cannabis. You smoke cannabis. So the entire situation is going to change here. So before we only had these ligands present, now we're introducing THC to our CB1 receptor, okay? When THC binds to our receptor, it causes what's called a conformational change. So this receptor will physically shift in the way that it's positioned. This tail here is going to cause a change, which is going to cause GDP to leave and GTP to bind to this subunit here. Here's where it gets into the downstream effects. So simply THC binding has caused GDP to leave, GTP to come. Then your alpha subunit is going to dissociate from your other subunits. So you have your alpha, and these are just kind of floating along the membrane. You have your alpha subunit. And your beta and gamma subunits. These can then go and interact with other membrane proteins. And eventually, these are going to cause a chain reaction in our body to cause, so one ligand binding will cause a huge downstream effect that will have other physiological functions. You might activate cyclic AMP, which is going to cause your cells to just interact in a completely different way and essentially become activated via second messengers. So just to review the really important parts here, your ligands are binding on the outside of the cell. This causes a change in your GPCR conformation that's going to cause the G protein subunits to go cause other effects in the body. So outside of the body causes an inside of the body change. So our cannabinoid receptors within the endocannabinoid system, we have our CB1 receptor, or cannabinoid receptor 1, and we have our CB2 receptor, or cannabinoid receptor 2. There are other receptors that play a significant role in the endocannabinoid system, but we are going to focus on these two just to keep things simple for this review. These are both G protein coupled receptors. So they'll have the 7 transmembrane helices, which allows them to take a signal from outside of the cell and translate it to inside of the cell. As I mentioned, they're both part of the endocannabinoid system, but they are located primarily in different areas. So your CB1 receptor is mainly part of your central nervous system. So it's going to affect your brain chemistry. In contrast, your CB2 receptor is mainly in your peripheral nervous system. And that's going to affect things like your gut, your skeletal muscle, your immune system, etc. But even within your brain where your CB1 receptors are located, your brain has very distinct and diverse areas. So the actual physiological response that you will get from ingesting cannabis molecules, it's going to depend on where the CB1 receptors are actually located. So, for example, you have CB1 receptors in your cerebellum, your hippocampus, your basal ganglia, and these areas are involved in things like pain, in memory, short and long term, and your motor control. So you can see that when you were to ingest something like THC, these are going to affect these parts of the brains and cause these physiological effects. So just a review of your traditional neurotransmitter signaling, just so we can talk about how phytocannabinoids are different. So say this is your presynaptic neuron, and this is your postsynaptic neuron. So in normal neurotransmitter release, we have these little vesicles, which are going to combine on the outside here. They're going to release these neurotransmitters where they're going to hit a specific receptor on your postsynaptic neuron. We will find an action potential that started here. After activation here, it'll just continue down, and that's how we transmit these signals in the brain. But when we're talking about the endocannabinoid system, it's a little different because we are talking about retrograde signaling. And retrograde signaling is essentially, we have retrograde signaling because we're trying to maintain what's called homeostasis. So if something gets a little bit out of control, we're using the endocannabinoid system to kind of reset our body or turn off certain valves so that certain neuronal processes aren't getting out of control. So the endocannabinoid system needs to react really quickly. It needs to happen exactly at the site of the neuron. So what does this mean? It means that we need the building blocks for our endocannabinoids to be present on the postsynaptic neuron so that we can immediately make them and then cause an effect on the presynaptic neuron. So how is this possible? I've drawn various lipid bilayers that looked something like this, and I'm sure you've seen these before, but these are what we're going to call our lipid bilayer, and it's what makes up the membranes in our body. So anything that is attracted to water is going to orient itself so the other things that are attracted to water are going to be close to the water. So here what we're really focusing on is these OH groups. So water is H2O, right? So when we see OH groups in our molecules, they're going to be the ones on the hydrophilic side of the molecule. Hydrophilic meaning likes water. The inside of this lipid bilayer is going to be composed of hydrocarbons. So a hydrocarbon is part of a molecule that's only hydrogens and carbons. You'll often see them kind of depicted like this, but remember that each of these dots are carbons. And then here we would actually have a bunch of hydrogens. They're just usually not drawn out. So again, hydrocarbon is a very fatty molecule, and a hydroxyl is not a fatty molecule. It does not contain carbon and hydrogens. It's oxygen and hydrogens. So this is what the middle is composed of, and then the outside is composed of this. But what if we were to use an example of what is actually making up our lipid bilayer? So what we're going to see that's making up our lipid bilayer is this molecule called arachidonic acid. And arachidonic acid has a carboxylic acid group, which we can see here, and is similar in structure to water. Remember, water's H2O? This is very similar to that. But what we're going to see is after this oxygen, it's going to become nothing like water. And we're going to see these kind of repeating. And again, it's a bilayer, so we would see the same thing here with this group pointing here, and then the lipophilic tail, and same thing. So again, you'll notice the carboxylic acid portion is going to be really similar to water. They're going to get along great. They like being around each other. In contrast, the lipophilic tails do not want to be around that water, so they're going to stick to the inside of the membrane. So if this is arachidonic acid and it is located in all the membranes in our body, it would be located on our neurons in our body, our axons and our dendrites. We're going to have arachidonic acid incorporated into the lipid bilayer here. So it's already present on the postsynaptic neuron. So when we have some sort of dysregulation of our neurotransmitters, say it's on a glutamic neuron, there's too much glutamate, it's too excited, then the enzymes in our body are able to immediately at the site where the issue is happening, say that our enzyme is, let's say this is our NAPE enzyme that is going to make our anandamide. So it is going to literally grab an arachidonic acid from that membrane, and the enzyme is going to turn the arachidonic acid into our anandamide. Right on the spot where it needs to happen, so it can immediately use that anandamide as a neurotransmitter. So we'll have NAPE enzymes there, and then we'll also have our DAG-L, alpha and beta enzymes right at the site of the neurotransmitter, so we can immediately grab that arachidonic acid and make these signaling molecules. So although I know that people aren't too familiar with looking at chemical structures, I do think reviewing anandamide, 2-AG, and arachidonic acid is really helpful for understanding where these molecules come from and why they're named what they're named. So again, I'm going to just draw arachidonic acid. Then I'll draw, then I'll draw anandamide. And then lastly, I will draw 2AG. Okay, so we can just visually looking at these three, we can tell that they look extremely similar from this part over. These molecules are the same if we're only looking at the right side of the molecule. Oops. But what's different is this part of the molecule. So on arachidonic acid, it's called an acid because we have a carboxylic acid. This is a functional group, and that's why that's called arachidonic acid. There is no double bond oxygen followed by an OH here or here. There is an O here, but it's not an OH, so that is not a carboxylic acid. When we talk about anandamide, we could also call it arachidonyl ethanolamide, or AEA, which seems like just a crazy name for this molecule, but it makes a ton of sense. So we just went over that the right side of the molecule is super similar to arachidonic acid, so that's where the arachidonyl part comes from. And then if we look at the ethanol part, that is this group here. So one carbon is a methyl, 2 carbons is an ethyl, and then OH is the ol. So that's ethanol. And then, so that's this part here. And then the last part here, amide. So that's another functional group here, which is a double bond oxygen next to an NH. Again, amide, not amine. If it was an amine, it would not have a double bond oxygen right next to it. It would just be an NH with just carbons next to it. So that's where that name comes from. And then if we look at 2AG, what we'll notice is we have a glycerol group here. So we notice there's a glycerol group here, and the actual name of 2AG would be 2-arachidonyl glycerol. So again, the right side of the molecule is the same as arachidonic acid, and then we have a glycerol functional group. So this is where the names actually come from, and this is where you can really tell how similar they are, and that's why they act in similar ways. However, enzymes are very similar to receptors. So they're so specific. So you need a specific enzyme to synthesize this one and a specific enzyme to synthesize this one. So that's why we have our NAPE enzyme and we have our DAG-L enzyme. They're able to make these different compounds, but they're still using arachidonic acid from the cell membrane as their starting material. Let's also talk about, sometimes you'll read about arachidonic acid and it will be referred to as a long chain polyunsaturated fatty acid, which is a long way of describing it, but long chain, okay, it's a long chain, it's like 20 carbons, but polyunsaturated, that's referring to this part right here. This row of double bonds, if this was a saturated molecule, it would simply look like this. And you could think of that as just being saturated in hydrogens. So there's no double bonds. So instead, at each of these places, there's hydrogens, etc. So that's just kind of another name to describe these molecules. Okay, so next we're going to review a video that Dr. Dankaz has a wonderful job explaining for these excitatory and these inhibitory neurons. So let's start with our glutamate as our signaling molecule. So again, this is going to be our postsynaptic neuron, and this is going to be our presynaptic neuron. Typically, we would see the vesicles that held the neurotransmitters in the presynaptic neuron go to the postsynaptic neuron and cause an action potential. That's not what we're going to focus on here. Instead, we have glutamate that in normal conditions would be exocytose. It's going to be here, and that's going to activate some sort of receptor and cause our signaling. This is still going on, but then our body says, all right, we're way too excited. We need to calm down. And remember that this neuron, its purpose is to be excited. It's an excitatory neuron. That's its normal state, is to be excited. So if we want to maintain homeostasis, so our body says, okay, we're going to kind of turn down the valve a little bit here. We want to maintain homeostasis so we don't get sick or anything. That's when the enzymes that are already present here, so in this example, let's say it's the DAGL alpha, it's going to grab an arachidonic acid from the cell membrane, and it's going to synthesize 2-AG. So it was able to do that on the spot of this postsynaptic neuron to respond to the glutamate excitation. So this is very quick. It's grabbing it right from the membrane and causing response. It's going to send that 2-AG to the presynaptic neuron, where the 2-AG will bind to our CB1 receptor. Then it will cause a biological response. It will decrease the amount of glutamate released, which is ultimately going to decrease the excitation response or cause less action potential. After the 2-AG has done its job, it's actually going to just float back here, where it will be degraded by the enzyme responsible for degrading 2-AG. Again, that's a very specific interaction. So for 2-AG, that's the MYGL enzyme that's going to recycle the arachidonic acid back to the membranes, so it can always reutilize these molecules. Just to review, if you are inhibiting an excitatory neuron, you're overall decreasing the activity of this neuron. And let's talk about the other example. Ooh, that's ugly. All right, so now we're talking about GABA as our neurotransmitter. And remember, the goal of GABA, its purpose is to be an inhibitory neurotransmitter. So that's its biological function, right? To inhibit. So if it's typically inhibiting, GABA is going to activate a calcium pump, which is going to cause so much of a calcium influx that nothing's going to happen. This receptor is just inhibiting its normal function is to not function as an excitatory neuron. But if we say, our body says, oh, actually, like we're inhibiting too much, we definitely want to excite a little bit to cause some sort of response, then again, our enzyme says, yeah, no problem, I'm already right here, and arachidonic acid is already right here. So in this case, let's say that this is the NAPE enzyme. It's going to grab its arachidonic acid, and it is able to really quickly make the, I did 2-AG. Let's say it's an anandamide. and it can immediately send an anandamide to the presynaptic neuron, where it will then again bind to your CB1 receptor, cause less GABA to be produced, which will cause less of an inhibition, which will overall result in excitation of this neurotransmitter. And then we will see the action potential actually make its way through. And again, that anandamide would then kind of just float back across the membrane, where it would be degraded by a specific enzyme. So this is the FAAH enzyme, and then the parts would be recycled to use again as part of the lipid bilayer. Okay, so now that we have a basic understanding of some of the pharmacology happening within the endocannabinoid system, let's talk about cannabis. So we have two main players when we're talking about the molecules in cannabis that are going to cause some sort of response in our body. And I'm sure we're all familiar with these, but let's draw them out. So we have cannabidiol, or CBD. And then we have our THC. And remember, these structures that we're used to seeing are actually the decarboxylated versions of the molecule. So when the plant is making the molecule, it actually is adding in a carboxylic acid at this position on both compounds. We need to heat up these compounds or the plant material to a specific temperature so that these leave in the form of CO2, and we're left with just the structures in the black. So remember, when that carboxylic acid functional group is present, that gives us a different key. And this key does not fit one of the receptors we care about. So we need to change the key to be able to activate the receptor. So once we decarboxylate our molecule, CBD and THC are still different in a very important way. THC has a ring structure in this position where the oxygen is incorporated into the ring, and CBD has a hydroxyl group, or this OH group here, which is not part of a ring. The rest of the molecule is identical, but remember, because this contains a cyclic group and this one doesn't, their keys are different. So maybe CBDs would look like this, and THCs would look like this. But we know that these are going to activate different locks, aka different receptors. Some things that these molecules have in common is their molecular formula, C21H30O2, and they have the same molecular weight of 314 Daltons. So the reason that THC can bind to the same receptor as our endogenous ligands, such as anandamide or 2-AG, is because it contains a similar pharmacophore. So when I say pharmacophore, we're really talking about a part of the molecular structure that's responsible for interacting with the receptor and causing some sort of activity within our body. So I'm going to show an image that was published in a journal called Nature, and it kind of just highlights what a pharmacophore is. Okay, it's a little blurry, but I think we can get the gist out of this. So on the left, we have THC, and on the right, we have anandamide. At the bottom right of each molecule is a 5-carbon chain, a saturated carbon chain, so just carbons and just hydrogens. And this saturated part is interacting with the receptor in such a specific way that it causes the receptor to become activated. Similarly, we can see that the tops of the rings, so the methyl group for THC and then the hydroxyl group, those are highlighted as important parts for activating this receptor. And then similarly, the carbonyl group and the hydroxyl group are also important for activating this receptor. So when we say pharmacophore, we're really talking about these parts of the molecule that are highlighted in this example. However, ligands, or small molecules like CBD, THC, anandamide, and 2-AG, they can interact with receptors in a few different ways. So again, I'm drawing my lipid bilayer because this is a transmembrane receptor. And first, we're going to talk about agonist molecules. So an agonist molecule, there's a specific part on the receptor where that agonist is going to bind. So if the agonist is present, it's going to bind in that specific location, and it's going to cause a biological response. And this site is typically where the endogenous molecule would also bind. So for example, if anandamide bound to the specific location and caused an agonist response and some biological response, that's also going to be where THC would bind as an agonist and cause a similar response. You could also have full agonists, which are going to cause the complete response possible for that receptor. You can have a partial agonist, which is going to cause not the full response, but some of that biological response. And then you could have an inverse agonist, which gets a little more complicated. But the second example we can talk about is an antagonist. This looks a lot like a baseball. That wasn't on purpose. Okay. So when we're dealing with an antagonist, it's going to bind, even though it's not that molecule, it's going to bind in that same location as the agonist would. However, it is not going to cause a biological response. And it's also going to prevent the agonist molecules from binding and producing a response. So in this case, we have a molecule bound at that active site, but it's not the right key for an agonist response. So the agonist can't bind. And then our last example is going to be called an allosteric modulator. So here again, we have the site that we know things like THC and anandamide can bind to. But nearby, we have a different site where a different molecule can bind to. And say that this is kind of a bulky molecule. So when you have a molecule that had bound to this other site, and that's preventing your agonist molecule from actually reaching anywhere because it can't physically reach a spot where it needs to. So this is a receptor location in a different location, and it will likely decrease the activity of an agonist because it just can't get there. So in this example, this triangle here, this would be CBD, which acts as an allosteric modulator. And then this would be your THC, who's just trying to get to the active site here, but it can't. So in that way, CBD kind of works with THC to help not cause too much of a biological response, which can activate adverse reactions, and it can make you feel some of the not great things from cannabis products. The last topic that we're going to really discuss is going to be going over metabolism. So let's just review from our molecular formula of THC and CBD of C21H30O2. This is mostly carbons and hydrogens with just a couple oxygens. So it's mostly a hydrocarbon, which we know are the lipophilic parts of molecules, and then just a couple oxygen. So these are lipophilic molecules, and for that reason, when our body has too many of these metabolites present, it stores them in adipose tissue. And adipose also means fat. So this is one of the reasons that these cannabinoids will stay in your body longer than many other drugs that could be excreted in something like the urine, these water-soluble molecules. The fat-soluble molecules are just kind of stored in the fat throughout your body. So we've mentioned the effects of vaping or smoking, and in that way it's kind of going right to your bloodstream and your brain. But what if we were to orally ingest these molecules? So what if we were to eat them? Edibles. We have a completely different metabolism when we eat cannabis products versus when we smoke or vaporize them. This is because we eat them and then they go through our digestive tract and then they're exposed to first pass metabolism in the liver. So the liver has special cells that are specialized in metabolism. So they contain an enzyme called cytochrome P450 enzymes, which are assisting in the breakdown of compounds. So often you're adding certain groups that are going to make it easier for your body to excrete these compounds later on. So in the liver, we have, say we took a product via, orally, and it's going to cause some effect in our liver. So again, this is our THC, our main psychoactive compound, or delta-9 THC, as you'll see it referred to often. Again, it went through our digestive tract. It is now interacting with these cytochrome P450 enzymes. And what do these enzymes do? They don't make a lot of changes, but they make a really important change. So before, we had just this little methyl group up here. Now we have that methyl, but we've attached a hydroxyl group, this OH group. And that changes how our body reacts to it and how this molecule interacts with our CB1 receptors. Because remember, our key has changed now. And this has a greater affinity for those CB1 receptors, so it's going to cause more of a psychoactive response. you don't always want a crazy psychoactive response, especially when you're taking cannabis for medicinal benefits. You don't want to feel like you're seeing things and kind of going crazy. So this is not an effect that a lot of people want. And a way to prevent the production of 11-hydroxy THC is to take a cannabis strain that also contains CBD. Again, the non-psychoactive cannabinoid in cannabis. So, CBD is able to inhibit your cytochrome P450 enzyme. And when you inhibit this enzyme, there's nothing there to convert THC to 11-hydroxy THC. So this product is either not produced or not produced as much. Another benefit of taking cannabis products that contain CBD is CBD is an agonist for what's called the 5-HT receptor. This may just sound like a random receptor, but it's not. It's actually our serotonin receptor. So CBD acts as a 5-HT receptor agonist, and your 5-HT receptors are also G protein coupled receptors, you guessed it. So this, when we have more serotonin produced, because this is an agonist, makes us feel happy. It also will relieve anxiety symptoms due to deficient serotonin in our bodies. So conclusively here, combining CBD with THC can really prevent some of the adverse effects seen by too much THC or too much 11-hydroxy THC. I hope that this review has helped in some way. Please let me know if there's anything else that you want me to review. I'm happy to make these videos whenever you would like. And again, my email is rileycirk@uri.edu. You can email me anytime you want. Thank you for tuning in, and I hope you enjoyed this video.